Hello everyone. Today we are going to discuss about endoplasmic reticulum, vacuole and Golgi body. Endoplasmic reticulum is a vast network of membrane-bound, branching and interconnecting tubules, vesicles, irregularly distributed in the cytoplasmic matrix. It is divided into rough endoplasmic reticulum and smooth endoplasmic reticulum. Now we can see the major differences between rough endoplasmic reticulum and smooth endoplasmic reticulum. In rough endoplasmic reticulum, ribosome is present, composed of a network of flattened sacs called cisternae. Location for the synthesis of protein constituents of endoplasmic reticulum may develop from nuclear membrane, abundantly found in exocrine pancreatic cells and antibody secreting plasmid cells. Pores are present. Coming to smooth endoplasmic reticulum, ribosome is absent, mainly composed of tubules. Synthesis of non-protein substances takes place, may develop from rough endoplasmic reticulum, abundantly found in liver and testicular cells that synthesize steroids. This is the picture of endoplasmic reticulum. Here we can see smooth endoplasmic reticulum, ribosomes, nucleolus, nucleus, etc. Endoplasmic reticulum exists in three morphologically different forms, that is, lamellae, vesicles, and tubules. Lamellae are unbranched and flattened sacs arranged in parallel stacks. Their internal lumen is cisternae. Vesicles are closed, oval, or rounded sacs. Tubules are branching and interconnecting units. Lamellae are granular, as well as vesicles, tubules are agranular. When cells are homogenized, their endoplasmic reticulum may get fragmented. These fragments soon get reorganized, forming small vesicles called microsomes. Here we can see lamellae, vesicles, and tubules. Coming to the chemical makeup and molecular organization, membranes of endoplasmic reticulum are formed mostly of phospholipids, cholesterol, intrinsic and extrinsic proteins. Phospholipids forms a bilayer and cholesterol binds with this bilayer to form a stable complex. Intrinsic proteins partially or completely penetrate the lipid bilayer, whereas extrinsic proteins remain entirely outside the lipid bilayer. Two characteristic transmembrane structural glycoproteins in the are ribophorins 1 and 2. They are absent in smooth endoplasmic reticulum. They serve as the binding sites for ribosomes and also mediate the binding of ribosome with rough endoplasmic reticulum. The functions of smooth endoplasmic reticulum are the synthesis of steroid hormones in the endocrine cells of the gonad and adrenal cortex, major role in detoxification, sequestering calcium ions within the cytoplasm of cells. The functions of rough endoplasmic reticulum are protein synthesis, transport of proteins, transport of nucleus material to other organs. Sarcoplasmic reticulum is an extensive network of specialized smooth endoplasmic reticulum found in striated muscles. It is concerned with the storage of calcium 2+ ions. Calcium 2+ ions are stored in the endoplasmic reticulum with the help of calcium binding proteins. Next, we can move on to Golgi body. Camillo Golgi was inventing new types of staining procedures. Camillo gives metallic stain to nerve cells from the cerebellum and discovered a darkly stained reticular network located near the cell nucleus. It was Golgi complex. He got Nobel Prize for the invention of Golgi body. Golgi bodies are aggregations of membrane-bound spaces particularly abundant in secretory cells. This is Camillo Golgi. Next, we can see the functions of Golgi body. Golgi body serve as an intracellular compartment for the chemical modification and transformation of substances. It synthesizes mucopolysaccharides and glycoproteins. It also secretes mucus, milk protein, thyroxine compounds. Golgi body consists of flattened disc-like membranous cisternae with dilated rims, associated vesicles and tubules. The diameter of cisternae is 0.5 to 1 micrometer. We can see almost 8 cisternae in the Golgi body. Golgi body contains 3 structures: lamellae, vesicles and vacuole. Lamellae form the core unit of Golgi body. Vesicles and vacuoles remain crowded around them in clusters. Here we can see vacuoles, lamellae and vesicles. Each lamellae has a polarized orientation. It has a convex outer side or face known as the forming face, otherwise known as entry face or cis face. It also has a concave inner face known as maturing face. It is also known as exit phase or trans phase. Each phase is closely associated with a network of tubules forming cis-Golgi network and trans-Golgi network. Next we can move on to vacuole. Vacuoles are fluid-filled and membrane-bound intracellular compartments. First we can see plant vacuole. The membrane that bounds the vacuole is known as tonoplast. We can see pigments called anthocyans in vacuole. They are red, blue or purple in color. It is responsible for the colors in fruits, buds and leaves. In the meristematic cells of plants, vacuoles are either absent or extremely small. In this picture of vacuole, we can see tonoplast, central vacuole and cell sap. The fluid that fills a plant vacuole is called cell sap or vacuole sap. The osmotic entry of water into the cytoplasm from outside and also to the cell sap from the cytoplasm builds up a pressure potential within the cell known as turgor pressure. In return, the cell will exert an equal and opposite counter pressure known as wall pressure. Now let's see protist and animal vacuole. It includes phagocytic vacuole, autophagic vacuole, food vacuole and contractile vacuole. Contractile vacuoles are absent in animal cells. It is osmoregulatory in function, that is, maintenance of osmotic pressure. Intracellular digestion is the function of phagocytic vacuole, autophagic vacuole, and food vacuole. These are the functions of vacuole. Cell solutes and macromolecules, including ions, sugars, are stored temporarily in the vacuole. Vacuole is the host of toxic compounds. It also isolates byproducts from metabolic reactions. This is all about endoplasmic reticulum, vacuole and Golgi body. Thank you everyone.